Hello, everyone. My name is Laura Ann Lowery. I'm an associate professor at Boston University School of Medicine in the Department of Medicine in section of Hematology and Medical Oncology. I am very grateful for this opportunity to tell you about the research in my lab, and so I want to thank the organizers of this conference for inviting me to speak to you today. So the big picture research question that has fascinated me in my lab for as long as I can remember is, how do cells move? How do they find their way? How does a cell move towards the right direction, whatever that may be, and away from the wrong direction? Now, this is a fundamental question in cell biology. Cell movement happens in the embryo during development. It happens in the adult during wound healing and the immune response, and it happens inappropriately during cancer metastasis. Errors involving cell movement can have disastrous consequences with a variety of pathological situations. And this is especially true for cell movement during embryonic development. Constructing the embryo involves an elaborate orchestration of both collective and individual cell movements. Many different cell types have to migrate over a complex embryonic terrain to get to where they need to be for proper tissue assembly and organ formation. The developing nervous system, which is where most of my lab's research has been focused. Neurons have at least two migration challenges. Each neuron first is born in a particular location and then has to migrate to a different location where then it is its home for the rest of the embryo's life, the rest of the organism's life. And then after this movement, after this migration process, each neuron then sends out an extension of itself called an axon and sends it over long embryonic distances to make those essential connections to wire up the nervous system with accuracy and precision. Now, defects in this process of connectivity lead to a long list of neurodevelopmental disorders, including, we now think, it contributes to autism and schizophrenia. Now, in forming these essential neural connections, the primary player in this process is the growth cone. This is the dynamic hand-like structure at the tip of the growing axon. It's the growth cone's job to find its target. My lab focuses on how it moves in the right direction. Now, this movement that the growth cone exhibits, any movement really, is a mechanical process. There is a machine inside these growth cones that propels them forward and steers in the right direction. And this machine is called the cytoskeleton. In primary embryonic growth cones, there are two major components of the growth cone cytoskeleton. There's actin, which drives motility, movement of the growth cone, and then there are microtubules, which provide the organization of the growth cone and allow for steering. Now, my lab focuses on how this machine is controlled, which is a long-standing question in the growth cone cytoskeletal field. And what my lab uses is the model system Xenopus laevis to tackle this question. With this system, we can easily inject constructs for gene manipulation into the early embryos. The next day, we can dissect out the neural tubes, culture them on cover slips, and then the following day, axons have grown out. Here you can see the axons emanating from a neural tube explant. And if we zoom in to just a single growth cone, their large and impressive growth cones allow for clear visualization of individual microtubules, as well as complexes, protein complexes that are found on microtubules. There may be no other vertebrate model system that has primary embryonic growth cones as large and as easy and inexpensive to culture, manipulate, and image as Xenopus laevis. And that's why I chose this model system for my lab's work. In my lab, we study a number of different projects related to cell movement. So I'm going to just give you a brief overview of them. Now, as I mentioned, the major focus of my lab's work is studying the growth cone. And in this context, we study microtubule regulators and how they play a role in axon guidance. And again, this has been the major focus of my research. I'm going to talk about two different vignettes within this topic after the introduction. But my lab is also diversified and looking at other different research areas that are related. So we also now look at neural crest cell migration within the context of a human disorder called Wolf-Hirschhorn Syndrome, which I'm going to talk about at the end of this talk. And we're also now using Xenopus laevis neural crest cells as a model to study cancer metastasis. Finally, we are also using Xenopus laevis as a rather inexpensive medium throughput system to identify genes, the functions of genes associated with various human intellectual disability disorders. So this is a broad range of topics all related to either cell movement or neural development. And today I'm going to talk about the microtubule regulators and axon guidance and Wolf-Hirschhorn syndrome. So first, let's go back to this growth cone, this dynamic structure at the tip of the growing axon. So I mentioned it's the growth cone's job to find its target. It needs to make decisions, whether to move forward, to retract, or to turn. And I told you this movement is a mechanical process, and so there is a machine called the cytoskeleton inside the growth cone. And in order for the growth cone cytoskeleton machine to move in the correct direction, there have to be various cues that the growth cone encounters, both negative guidance cues, positive guidance cues, that then are somehow translated to converge upon the cytoskeleton so that the cytoskeleton then grows in the right direction and moves the growth cone in one direction or another. So if we zoom in to a cartoon of this growth cone, you have red actin filaments in this cartoon in the periphery, and again, they're driving growth cone motility. And then there are microtubules in blue, which both form the core of the axon shaft, as well as there's dynamic microtubules that explore the growth cone periphery. They even go into these filopodia, move along F-actin bundles, and these microtubules can perform this internal growth cone exploration by utilizing their property of dynamic instability, where microtubules switch between growing phases and shrinking phases. And by rapidly switching between these two states, they can effectively explore the entire growth cone periphery and even go into those filopodia. And that's where the guidance cues are first going to be detected. Now, on the tips of these microtubules, there are a series of proteins called plus N tracking proteins, or plus tips. Here in this microscopy image, you can have the plus tip in green with microtubules in red, and you can see it forms these comet-shaped accumulations on the ends of the microtubules. Now, plus tip activity is directly modulated by upstream kinase signaling. Not only do plus tips bind to and regulate microtubules, but they also interact with structures such as F-actin and cell adhesions. And so it seems quite plausible that plus tips might take a central role in orchestrating the growth cone response to guidance cues, but the details for how this might occur are largely unknown, as plus tip interaction and function within the nervous system has not been extensively examined, and so that's where my lab came in. So back when I was still a postdoc at Harvard Medical School, I did a screen to identify new microtubule regulators that could be plus tips that might be playing a role within the growth cone to promote axon guidance. It was a very fruitful screen. I used Drosophila. It's a great screening platform. Identified a number of potential microtubule regulators. The two that I'm going to be focused on for this talk are XmAb215 and TACC3. It's been known for over a decade that these two proteins bind to each other and interact to regulate cell division, but there was little known about their functions during neural development when I studied them. XmAb215 is also called ChTOG in humans, or CCAP5. Now, XmAb215, a lot was known about this, decades of work. XmAb was known as the classic microtubule polymerase. It would bind tubulin dimers in the cytoplasm, as well as the end of microtubules, the plus end, and it would deposit those tubulin dimers onto the plus end to promote microtubule growth. Thus, it functioned as a catalytic microtubule polymerase. It was also highly enriched at the centrosome from where most microtubules nucleate. And it's thought that it's there so that XmAb215 could then load on to new microtubules to promote their growth. What I'm going to tell you about today, though, is a new function for XmAb215, where it is more than simply a microtubule polymerase. Now, this work was spearheaded by a postdoc in my lab, Paula Slater, as well as a graduate student, Garrett Camarada, and they had the help of two undergrads, Annika and Ali. When Paula joined my lab, she wanted to explore an unexpected finding for XMAP215 knockdown growth cones. So I found that when we knocked down XMAP215, this catalytic microtubule polymerase, but only knocked it down partially, like 50%, we didn't get rid of it completely, just partially, we saw that the growth cones were larger. Now, this made little sense. So in this image, you can see the green is actin, which forms like the size of the growth cone, and red are the microtubules. And when Paula measured these growth cones, she found that when XMAP215 was partially knocked down, the growth cones were larger and the filopodia were longer. And she could rescue this by adding back full-length XMAP215, suggesting that this defect was specific to XMAP215 knockdown. This made little sense why knocking down a microtubule polymerase would lead to larger growth cones, longer filopodia filled with actin. We wanted to know, well, could XMAP215 be somehow affecting F-actin? Now, in a movie that I had taken, you could see the still image where XMAP215, this is a growth cone, you could see that it forms these puncta at the ends of microtubules, but also you could see it along the microtubule lattice, the side of the microtubule. This led us to wonder, well, in order for microtubules to grow along these F-actin bundles, the F-actin is also undergoing a process called retrograde flow. You can see this in the bottom left cartoon, where actin is flowing backwards towards the center of the growth cone, but the microtubules are moving forward. But if the microtubules are going to be moving along the F-actin, using them as guides, there has to be transient, frequent coupling and uncoupling, regulation of the interaction between microtubules and F-actin. And we wondered, could XMAP215 be playing a role in this interaction? So Paula looked at the alignment of the microtubules to these F-actin bundles, and she found that with the XMAP215 knockdown condition, there was a reduction in the percentage of microtubules that were aligned to F-actin. Again, this could be rescued by adding back XMAP215. And if she overexpressed XMAP215, there was an increase in the number of microtubules that were aligned to the F-actin. And so this suggested that, yes, indeed, XMAP215 can affect microtubule alignment with F-actin in growth cones. So she wanted to know, well, what part of XMAP215 was facilitating this interaction? So this is a cartoon of XMAP. It's a big protein. It's 215 kilodaltons. There's a number of domains. There are 5 TACC domains. TACC stands for tumor overexpressed gene. And there's a C-term domain. And so she made a series of deletion constructs to see which domains could rescue the XMAP215 knockdown microtubule F-actin alignment defect. And here's a busy graph. What you can see from this is, I'm going to focus your attention on, this is what it looks like when the full-length XMAP215 is expressed. There's a full rescue of the alignment defect. But when she looked at all these various constructs, none of them could rescue like the full length except for this TOG 1 to 5. So TOG 1 to 4 was not sufficient. And so the difference between TOG 1 to 4 and TOG 1 to 5 is not only the 5th TOG domain, but also this microtubule lattice binding region. And that led us to wonder if perhaps something in this region is allowing the microtubules to interact with F-actin through binding to XMAP215 on this lattice. But we wanted to know, this was the very first time XMAP215 had been associated with F-actin. So can it actually bind to F-actin in vitro? Is this even possible? So to test this, Garrett, my PhD student, used an F-actin sedimentation assay, where he combined polymerized F-actin along with purified XMAP215 protein, and then combined them together, spun them down. Anything that bound to the pellet was binding to F-actin, and anything in the supernatant was not binding to F-actin. And what he found was that indeed, XMAP215 could directly bind to F-actin in vitro. You can see as the levels of F-actin go up, there's increasing levels of XMAP215 in the pellet. Okay, and this shows very strong binding to F-actin. And so this is the full length, and so of course the next question is, well, what domains of XMAP215 facilitate this interaction with F-actin? And so, again, we did deletion construct analysis. The C-term domain was not sufficient to bind to F-actin. You can see there's almost no XMAP215 C-term in the pellet in this image. The TOG1 to 4 was not sufficient for strong binding to the F-actin. Very little is in the pellet. But the TOG1 to 5, the same construct that could rescue the microtubule F-actin alignment defect, was sufficient to bind to F-actin in vitro. And so what I've shown you, and so you can see this nice binding curve. So what I've shown you in terms of the new function for XMAP215, that partial XMAP215 knockdown leads to larger growth cones. And to be honest, we still don't understand why the growth cones are larger. Okay, we need to look into that more deeply. But what this phenotype suggested for us is that F-actin could be playing a role. And so then we went further to determine that XMAP215 could promote microtubule F-actin in growth cones. We then also determined that XMAP215 can directly bind to F-actin in vitro, and this is facilitated by the TOG1 to 5 domain. We're still working on XMAP215, trying to figure out what it's doing and how it's regulated. But at this time, I'm going to turn to a related story on TACC3. TACC3 is a primary interactor of XMAP215. And when we first started studying it, first I just want to mention this work on TACC3 was spearheaded by my PhD student, Burju, and she had the support of 2 undergrads, Ben and Eric. So when we first started studying TACC3 was not in the literature as a plus tip. It was a centrosome adapter, and its main microtubule function was to bring XMAP215, the polymerase of microtubules, to centrosomes. Like, its main function, according to the literature, was just as a recruitment factor. But we wondered if maybe TACC3 could be acting as a direct plus tip, and it could be directly regulating microtubule plus end dynamics, more directly than simply recruiting XMAP. So we first just expressed GFP tagged TACC3 and saw that indeed TACC3 could be functioning as a plus tip. Here you can see a montage in the bottom of a single microtubule where TACC3 in green is at the tip of the growing microtubule. And we found that manipulating TACC3 function leads to changes in microtubule growth velocity. So when we partially knock down TACC3, there's a reduction in the velocity of microtubule growth. And when we overexpress TACC3, there is a slight increase in the velocity of microtubule growth. The lifetime of the microtubules were not affected, so the catastrophe frequency wasn't affected, just the polymerization rate. Now, consistently with this effect on microtubules, Burju found that when she reduced TACC3 levels, there was a reduction in the length of the axons that grew out. When there was an increase in TACC3 levels, there was an increase in the axon outgrowth. And this was all expected for a microtubule polymerase promoter. What was fascinating, though, is when Burju looked at time-lapse imaging, she saw that overexpressing TACC3 led to a mild reduction in axon outgrowth velocity. And this made no sense. If we are overexpressing a microtubule polymerase and the axons end up being longer, why would they grow out more slowly? How could that be? When she looked at the data more closely, she noticed that in control situations, axons growing on laminin-coated cover slips in vitro, they undergo spontaneous retraction at a certain frequency. And when TACC3 was overexpressed, the axons reduced their spontaneous retraction rates. Thus, these axons, even though they were growing more slowly, they grew more persistently over time, and that is how they ended up having longer axons. So this was interesting to us, and we wanted to know more about whether TACC3 could be mediating axon outgrowth in response to guidance cues. So what Burju did is she applied SLIT2, which is normally a repulsive factor, and she found that normally when you add SLIT2 to the media of these axons, there's a collapse of the growth. And you can't see it, the movie isn't playing, but if you were watching the movie, you'd see the axon would be collapsing. And what she found is that when TACC3 was overexpressed, there was a reduction in the growth cone collapse phenotype. Thus, this data demonstrated to us that TACC3 was somehow mediating the growth cone response to guidance cues such as SLIT. We wanted to know more about the signaling pathway involved. And so the first candidate we looked at was Abl-induced tyrosine kinase. It's known that Abl mediates SLIT signaling. We wanted to know, could Abl-induced tyrosine kinase be indirectly or directly phosphorylating TACC3? And so to do this work, we joined forces with a collaborator of mine, Brian Balef, at the University of Vermont. He is a mass spec and kinase expert. What we found is that when we expressed TACC3 GFP as well as Abl-induced tyrosine kinase in HEC293 cells, there was this really nice phosphotyrosine signal. We sent that off for mass spec. There were a number of candidates, a number of tyrosines that were phosphorylated, both in the TACC domain as well as the highly conserved regulatory domain. And so we wanted to know if we knock those out, could it affect the function of TACC3? When she knocked it out, first she did a Western and surprisingly saw that the phosphotyrosine signal was not changed at all. So then she knocked out the other possible tyrosines that could be phosphorylated, did a Western again after expressing them. And again, there was still a phosphotyrosine signal, which again was surprising. So then she decided, well, there's other tyrosines in TACC3. Maybe they're being phosphorylated, when some of the tyrosines are missing. So then she decided to focus on just the TACC domain, which is sufficient to drive polymerization, and there are 6 tyrosines there. So she made phosphonulls all 6. Again, she did the expression analysis and the Western, and she saw, surprisingly, that there was still a phosphotyrosine signal, despite the fact that there were no tyrosines left in TACC. But then she realized that we were expressing GFP TACC, not just TACC. GFP also has tyrosines. So when she expressed just GFP alone, she found that when Abl was co-expressed, GFP could be phosphorylated. Now, this is something that's really important for me to share with you because I didn't realize that GFP could be phosphorylated, and it isn't in the literature anywhere. So this is something that people should keep in mind if they're doing these sorts of experiments. But we still had this TACC phosphonull, and so she wanted to know, does the phosphonull change TACC's function? And here what she did was she expressed TACC, both the wild-type TACC as well as TACC phosphonull, and she found that under control conditions, wild-type TACC, most of it bound to the microtubule, but the TACC phosphonull, most of it did not bind the microtubule, and instead was stuck in the cytoplasm. Okay, so to summarize what I've shared for you about new TACC3 function. First, TACC3 binds to microtubules to promote their polymerization. Too much TACC3 leads to microtubule growth despite the repulsive cues and reduced axon retraction, and thus leading to longer axons. And that the TACC phosphonull cannot bind as well to microtubules. So kinase signaling does indeed affect TACC's ability to bind to microtubules and regulate axon guidance. And future studies in my lab are continuing to dissect the mechanisms by which TACC3 is regulated during growth cone guidance. Now, in the last few minutes of my talk, I want to turn to another area of research in my lab, looking at Wolf-Hirschhorn Syndrome. Now, this relates to neural crest cells, so I'm going to give you a brief introduction to them. Neural crest cells are a multipotent migratory embryonic cell type that delaminate from the neural tube during development, and they migrate throughout the embryo and become craniofacial structures, heart, skeletal, muscle, peripheral neurons, many different cell types. And this work was spearheaded by a former PhD student in my lab, Beth Beers. Now, we became interested in this disorder, Wolf-Hirschhorn Syndrome, when we realized that TACC3, which we were studying in the context of axon guidance for some time, we learned that TACC3 was also deleted in this human disorder called Wolf-Hirschhorn Syndrome, or WHS. There's a number of genes missing in this disorder. It's a hemizygous mutation. And the embryonic function of most of these genes was unknown. The defects these children have are craniofacial, heart, skeletal, cognitive function defects. A number of these defects could involve a defect in neural crest cell migration. So we wondered if it could be a neural crest cell migration disorder. So we decided to test that by looking at the functions of these different genes, the expression and the effect on knockdown. So first, we found that most of these associated genes are expressed, at least in part, in neural crest cells. And so here on the left, you can see a neural crest cell marker, and it stains these pharyngeal arches on the side with these, you can see it pointed to with the arrowheads. And we're looking at the side views of these heads of these Xenopus embryos. And the big blob that you can see are the eyes. And they're these pharyngeal arches, which form various craniofacial structures on the side. And most of these genes are at least partially expressed in these pharyngeal arches, suggesting they are expressed in the right place to be playing a role in neural crest cells. When we knocked each of them down individually, we found that they also knockdown impacted craniofacial morphology. Facial width and midface area are here, but they affected other parameters as well. We also found that they could affect the migration of the pharyngeal arches. So when we only knock down one side of the embryo with either WHS1 or TACC3, you can see that the pharyngeal arches are reduced in size. And we wondered, could it be because the pharyngeal arches the cells can't migrate as far? So to test this, we dissected out the neural crest cells and plated them and then measured motility parameters. We found that indeed knocking down WHSC1 reduces neural crest cell motility in vitro, as well as knocking down TACC3. We did a number of other assays, which I don't have time to talk about today, but what we found is that numerous genes in this region contribute to various aspects of craniofacial patterning. So all of them were expressed in the neural crest cells. When we knock all of them down, we see various defects in facial morphology. We see defects in cartilage morphology when we knock down 2 of them. When we see defects in pharyngeal arch length and neural crest cell migration when we knock down 2 of them. And then we also see defects in forebrain size. So they contribute to different aspects of craniofacial patterning. And continued work in my lab is now investigating, well, how does TACC3 specifically affect neural crest cell motility? But that's an answer for the future. So to summarize what I've presented to you today. First, I hope that I've been able to convey that Xenopus laevis is a fantastic embryonic cell model system for study cell migration in various contexts. XmAb215 not only is a microtubule polymerase, but it also can couple or align microtubule growth cone microtubules with F-actin. TACC3 is a plus tip that promotes microtubule polymerization during axon outgrowth and guidance. I've also shown you work showing that TACC3 likely contributes to the WHS phenotype along with the other candidate genes. And the current focus of research in my lab moving forward is studying how TACC3 specifically promotes neural crest cell migration. And also, I didn't tell you about 2 projects looking at how collective cell movements are regulated, as well as we're now transitioning to using Xenopus as a model system to study in vivo melanoma metastasis. With that, I want to acknowledge those who contributed to this work. First, I have select lab alumni, Beth, Burju, Garrett, Paula, and Sangmuk. They all did most of the work that I showed you today. My current lab members, spearheaded by my lab manager, Mackenzie Holm, and many students in my lab. I am very fortunate to work with a number of great collaborators. The Xenopus community is indebted to the National Xenopus Resource as well as XenBase, which provides Xenopus resources for us. And I appreciate the funding that has supported all this work over the years. So I want to thank you very much for your attention. If you have questions for me, I definitely welcome you to email me at laLowry@bu.edu. Find my website online. And thank you for your attention.